So it's a great pleasure to have Jacob Appelbaum join us for our seminar speaker. So Jacob did his undergraduate at Amherst and then did an MD PhD at Yale, where he did a lot of work on sort of designing protein-based drugs. And then through the course of his clinical training has switched, as I'll mention, onto more cell-based therapies. After his MD PhD at Yale, he moved to UCSF to finish residency and then moved up to the University of Washington for fellowship in Hematology and Oncology. He's an adult physician, which, as you can learn from his title, he has actually, in some ways, seen the light and moved to pediatrics for all the things that we can learn about the immune system. So he's going to tell us about a particular type of significant cancer that affects both adults and kids. So, Jacob.
Lovely to come back, and I will say that this work was done by a very large group of people, some of whom are in the audience. And as many of you know, I was in Mike Jensen's lab in Seattle Children's Therapeutics for about 6 years, and then recently moved over to Fred Hutch as an assistant professor. And this work is more of sort of a valedictory of all the hard work that was done during that time. And so I just will say thank you up front to all the hard work of the colleagues that I got to work with while I was in Mike's lab and here at, well, I guess so first over in Olive Lab and then over now in Building Cure. Okay, so I'm going to talk a little bit about some AML trials, some challenges and trade-offs in AML therapy, immunoengineering, AML CARs, and some of the specific hurdles that those therapeutics have to overcome, and then some risk benefits of next-generation CARs, and maybe some future planning, sort of a pitch for what I think we could do going forward. I don't have any significant financial conflicts of interest, but I did perform research in a lab that was supported by 2seventy Bio. And views are my own. So last year, unfortunately, a little north of 20,000 patients developed acute myeloid leukemia in the United States. Of those, half are expected to die of their disease. We haven't made a whole lot of headway in treating this disease if you look at the mortality rate over time. So year of diagnosis versus year of death, those are essentially flat over the past 20 years, showing that our new therapies have not yet impacted the outcome of this disease. There is a large variation in AML mortality that has to do with the age of the patient. So patients over age 65 are much less likely to survive. You can see their survival is below 10%, whereas patients under age 50, their survival is north of 60%. There is two peaks of incidence, one in the pediatric population. There's a nadir around the late teenage years, and then a peak around age 66, 67, so in older adult years. If you look at the absolute change of the survival versus age, in younger patients, the slope is actually steeper. So we're making more improvements in younger patients than we are in older patients. And I will say that one differentiating factor is the use of antigen-targeted therapy, particularly in younger patients. So younger patients probably have AML that is curable by chemotherapy. We're delivering potent targeted chemotherapy to the leukemic stem cells in that population. This is a hypothesis, but and that may be why that slope is slightly improved. I will say that there aren't big stepwise increases, just reflecting the fact that these are processes that improve gradually over time. It's not like there is one discovery and now all of a sudden everybody's outcome is substantially better. Immunomodulation is a hallmark of AML. The hallmarks of cancer keep growing, so now there's 2 circles instead of 1. But I will just highlight the issue of avoiding immune destruction and tumor-promoting inflammation. And so I think there's increasing recognition that pro-growth signals alone are one mechanism that drives increased production of bone marrow cells and immature cells within the blood. So that's myeloproliferative disease, but that alone is insufficient to drive leukemogenesis. You need the simultaneous presence of pro-growth signals, inflammation, and resistance to immune surveillance in order to generate leukemia. And that probably has to be coupled with genomic instability so that there's a dynamic evolutionary component to AML. And that's one reason why relapse is the most common precursor to death from this disease. What are the challenges of applying engineered cell therapy to AML? Where there are very few recognized leukemia-specific antigens. So a leukemia-specific antigen is an antigen that's only on the leukemic cell and not on any other cells. There's leukemia-associated antigens, those antigens that are enriched on the leukemia cells but also present on other cells. As I said, there are a few of those, but they're shared with hematopoietic stem and progenitor cells, or HSCs. And so an anticipated consequence of targeting these leukemia-associated antigens is bone marrow hypoplasia or aplasia. And so dealing with that toxicity will be important. There's an entity called clonal hematopoiesis of indeterminate potential, or CHIP. So CHIP is where a single hematopoietic stem cell is responsible for a production of a large fraction of the cells circulating in the blood. And what's been learned from this disease is that the reason that CHIP evolves is that that cell exudes an inflammatory mix of cytokines. So it creates an inflammatory milieu that blocks the proliferation of the normal hematopoietic stem cells. And at the same time, that cell, that CHIP clone, is resistant to the inflammation that it engenders. So it's kind of like standing, on the ladder, putting your foot on the guy below you. It's both blocking his growth and itself is resistant to it. And so that is a model for leukemogenesis, in a way, linking inflammation to proliferation. The genomic stability also likely involves immune evasion, and this may be broad in the sense that it induces exhaustion of tumor-reactive T cells. This immune evasion or exhaustion phenotype is present at diagnosis, worsens with increasing number of failed treatment cycles, and can be restored following successful therapies. And Adam Lamble in the back is probably world's expert in this particular feature. His PNAS paper was excellent on that topic. So antigen targeting is likely to require concurrent immunomodulation to eradicate AML. Antigen targeting alone is probably unlikely to be sufficient. Michelle Satterline's group in 2017 published this paper in Cancer Cell, where they looked at the distribution of various putative leukemia-specific or leukemia-associated antigens and tried to find one. Now, if you contrast these for AML, which is in the box on the top, with CD19, which is a sort of a canonical leukemia-associated antigen in ALL, the leukemia that's most frequent in pediatric patients, you can see that there are a few lymphoid tissues, the appendix, lymph nodes, spleen, tonsils, that are highly enriched in CD19, and the rest are basically not detected. In contrast, if you look at CD33, there's CD33 expression in the lung, prostate, in the tonsil, and various other organs, skin, that creates some concern for off-target activity. But perhaps a more relevant view is this view from Science. The spring graphs show hematopoietic stem progenitor cells at the top of this differentiating hierarchy of bone marrow cells. The color of the dots is the amount of expression of the labeled antigen within that particular population. And you can see that CD33 is decreased, has some detectable expression within HSCs at the very top of that triangle, but is mostly expressed within the monocytes and monoblasts. Other putative antigens that have been investigated, CD123, highly enriched on plasmacytoid dendritic cells. CD34 is the canonical stem cell antigen, along with CD117, CIIT. But these targeting these antigens would very likely get rid of your HSCs and HSPCs and leave the patient without a functioning marrow. One can also consider the consequences of targeting various cell populations, such as monocytes and dendritic cells compared to lymphocytes, and so that may have an impact on the function of the T cells following that antigen target engagement. CD33 is a validated target for AML. the addition of an antibody-drug conjugate, gemtuzumab ozogamicin, is now standard across a large variety of therapies. This meta-analysis showed that there is a benefit, although, and this was a big deal because at the time it was the first drug approved for this disease in about 20 years. Although I will say that the degree of improvement is modest, and using different dosing strategies led to contradictory results. So when initially approved, Mylotarg was dosed at a supersaturating dose, and that led to toxicity. Using just undersaturating doses in fractionated additions actually improved the therapeutic window, and so then there was a benefit, and the drug was actually approved, withdrawn, and then reapproved following this dosing change. What's striking is that the cytogenetics show very different outcomes based on the risk profile of the disease, likely reflecting the fact that there are some diseases that are CD33 targetable or curable if you eliminate that cell population, whereas others that are relatively resistant. So if you look in the favorable risk group, that benefit of gemtuzumab is quite substantial, leading from 6 years or 5-year survival from 55 to actually 75%. So that's a big difference. The difference is detectable, but not as substantial for the intermediate cell population or cytogenetic risk population. But for the adverse risk population, those patients who have myelodysplastic-like leukemia, that benefit is not detected. No, sorry. In pediatric patients, there's a lot of data for gemtuzumab, particularly in the MLL rearranged leukemias. So this is a report from Pollard et al. That was given in ASH, and then Catherine Tarlock also reported the benefit of gemtuzumab in reducing relapse and improving disease-free survival. And there was some trade-off, as I mentioned before, in terms of the risk of this approach, because you can see on the right, TRM is treatment-related mortality, and so you can see that there is an increase in the toxicities of transplant when Mylotarg is incorporated. So principles of AML therapy: the risk of relapse vastly exceeds the risk of toxicity, and that remains the greatest threat to patients. Early aplasia is good, but late aplasia is bad. So you need to have a functioning bone marrow in order to be safe. But it's also the fact that when you have a highly inflammatory milieu, the bone marrow does not work very well. And so the presence of aplasia is likely confounded because those patients are more likely to have resistant AML that continues to be present in the bone marrow after therapy. I would say that chemotherapy alone, cytarabine, daunorubicin, with or without Mylotarg, has been pretty well optimized. And so we're not going to make a lot of headway in sort of tweaking the various doses of the standard chemotherapy regimens that we use. Decreasing induction intensity produces inferior outcomes, so you can't go more, can't go less. This is where we're going to be for a while in terms of the standard approaches. Okay, so with that, 10 minutes later, finally get to CARs. This audience knows what a CAR is. Leukapheresis, that's your starting material. Load in the CAR either with a retrovirus or a lentivirus. Electroporated genome delivery, not a thing yet. CRISPR, not a thing yet, but it's coming. The cells are expanded in various ways, either with or without IL-2, and then reinfused into patients following lymphodepleting chemotherapy. There's no doubt that CD19 CARs are effective for B lineage malignancies. We saw 2 years ago at ASH second-line approvals, which showed an overall survival benefit for patients by intention to treat for liso-cel above standard of care. So this means that CARs are going to continue to expand in the population of patients for which they are indicated. And now that we know that this product is safe and effective, there will probably be disease spread in the sense of, first it's ALL, and then it's very aggressive lymphomas, and then it's mantle cell, it's other types of lymphomas and CD19 diseases, and then can we use it for autoimmunity and other things where we think B cell pathologies are important. So I think targeting B cells with CD19 is a strategy that will evolve, and for all the reasons that you might use rituximab. Now you might think of using a CD19 CAR if you need a bigger, badder rituximab. Okay, so typically CARs are always on, so why logic gates? So in an acute infection, which is a scenario for which the immune system is built, there are pathogens. There is a naive pathogen-specific CD8 T cell. It sees an antigen in an inflammatory context. The antigen is cleared, and you get memory. In the case of tumorigenesis, that naive tumor-specific CD8 T cell engages potentially with a cancer-associated antigen in a non-inflammatory context, and that antigen persists. And that persistence plus context leads to dysfunction. And so that late dysfunction leads to a reduction in the effector functions in terms of cytolysis, granzyme B, interferon production, and upregulation of inhibitory receptors like PD-1. So with an acute infection, T cells develop memory phenotypes and exhibit recall. Within solid tumors and in leukemia, T cells develop exhaustion, suppressing recall responses. Okay. If you could turn the CAR on and off, you might be able to limit the experience of antigen persistence by the cell. You could prescribe certain durations of antigen exposure. You could limit reactivity during periods of toxicity. And potentially, if there was a degree rather than a qualitative relationship between reversibility, you could use an activating molecule as a rheostat to define a therapeutic window. I'd say this is probably likely to be very challenging in practice, but I think at least conceptually, you could understand a way of giving, of having the CAR be just active enough. Autonomous CARs, sort of typical CD19 CARs, don't allow a user to intervene post-development. The cell product expands within the patient. You can have toxicity, and that can vary cell to cell. There are examples of switches. The SYN notch is one that has been well described. This is challenging because you need to engineer a transcription factor that is here that is not a typical CAR. So you have the cytoplasmic domain that then has a transcription factor that then drives a second CAR. And so this CAR is not a normal CAR. So so that particular, and in fact is not, does not necessarily functionally activate the T cell. So there's a lot of engineering within SYN notch that gets you away from a standard CAR design in terms of its intracellular activity. There's another way which is called the SNP CAR, which might be a little bit more attractive. In this case, the CAR has a cleavable domain on the intracellular portion, and then there's a membrane tethered protease that has a specificity for this linker domain here, and that protease can be inhibited by antiretroviral drugs. So in this way, in the absence of the antiretroviral drug, the scissors are on, it keeps getting cut, and the CAR doesn't do anything. In the presence of the drug, the protease is inhibited, the CAR remains intact, and now it signals. There are CAR T cell trials for AML in the United States. This is a little bit old, but you can see that these are expanding. I will say that so far the signals have been weak, although the COG trial seems to have some early signs of activity. Hasn't been published yet, and I look forward to ASH this year, where we'll probably get yet another update on the promise of that particular approach. The other cases are really limited to case reports. Maybe I would say in most cases, 2, 3, 4 patients in individual cases. The UCART I think was up to about 15 patients there, 16 patients, 4 of 6 responded. You can see that in the UltraCAR, 3 of 11 patients responded. And then in the CHLA CAR, that's some responses at higher doses, but all needed transplant. Okay, so if you're thinking about a CD33 CAR, how do you go about this? One idea is, could you create a leukemia-specific antigen? And this was an idea that was put forth by Miriam Kim when she was at UPenn. And so the idea here is that you can take donor hematopoietic cells, gene edit them to remove CD33, and now these hematopoietic cells are immune to the CD33 CAR. The leukemia cells, on the other hand, are susceptible to the CD33 CAR and will remain targeted. So now you take your healthy donor, you make CD33 CARs, they target the leukemia, they don't target the gene-edited stem cells. You infuse both the CARs and the gene-edited stem cells into the patient. The CARs eliminate the AML, they don't touch the gene-edited stem cells, and now your patient is cured. Well, that's one idea, and Stargill is actively pursuing that, and we actually have that trial at the Hutch right now. So we'll see if it goes anywhere. But another approach is to create a controllable CAR system in which you can have a period of CAR activity and then pause. The approach to this is using an FKBP-FRBP hybridization domain, heterodimerization domain, and the CAR is split in half. So in this case, you have a CD33 CAR, you have a FKBP and a transmembrane domain, and then in a second transmembrane protein, you have the FRB attached to the 41BB zeta. At the resting state, these two pieces swim around separately on the cell surface. Add RAPA and the FKBP-FRB heterodimerize. That interaction is very strong, one of the strongest known in human biology. And that reconstitutes a functional CAR. This CAR is, we refer to as ready because by itself it's not on. It has to engage antigen in order to activate the T cells. If you wash away the rapamycin, eventually that complex will dissociate, the rapamycin will diffuse away, and now you're back to the resting state where even if this antigen targeting arm engages with the antigen, it doesn't recruit the signaling activation domain. It doesn't do anything. We refer to this architecture as DARIC, which is dimerizing agent regulated immunotherapy complex, and this allows for rapamycin controlled activity. So now I'll take you through briefly some of the work that we did to develop this CAR product. We screened several single domain anti-CD33 binders. So there's a llama somewhere down in California that got a shot and then had a blood draw and made hybridomas. And llama bodies are single domain and don't have to have heavy and light chain pairing like human antibodies do. So they're very stable, and it turned out that a high proportion of these llama bodies, when grafted onto the outside of a CAR, worked great. And I think like the hit rate was like 95 out of 97, so like ridiculously high. If you have to make a CAR, I recommend nanobodies. There's some differences in terms of the amount of interferon gamma, probably related to expression levels, but all of them showed RAPA-responsive interferon gamma induction when they were challenged with CD33 tumors. We used a binding assay where we took CD33 expressing cells and then doped in various concentrations of VHH protein to measure the affinity of the VHH protein to the CD33. And you can see that the clone in blue there had an affinity that was around single-digit nanomolar. I think it was like 7 nanomolar. The black clone had an affinity that was about 10 nanomolar, and then there is a low-affinity clone that was about 100 nanomolar. Interestingly, the 100 nanomolar binder also had a binding to a different domain, which is in CD33. So most of the extant CD33 antibodies bind to the membrane distal domain. That's the VSET domain. There's a CSET domain, which is membrane proximal, and it was thought that targeting that domain might be more beneficial. In addition, there's some data that because Mylotarg targets the gemtuzumab ozogamicin, the existing CD33 antibody drug conjugate, targets the membrane distal domain, there was some splice isoform variation that was identified, that not all the patients express that distal domain, at least at the mRNA level, and so targeting the membrane proximal level might provide a little bit more universality when we were bringing this to the clinic. We looked at the antigen sensitivity of the CARs by titrating in various amounts of antigen, by electroporating various amounts of mRNA into HEC293 cells and using those as tumor targets for cell product. So if you take the same effector T cell to target cell ratio, so this was 3:1 all the way across, and then doped in escalating amounts of CD33 mRNA, you get this curve, which is basically that the cells express increasing amounts of CD33 on their surface, the target cells. And then when you challenge those cells, this is with the Derek T cells, this is the amount of interferon gamma that you get in the presence of RAP in the filled circles and the absence of RAP in the empty circles. And note the scale here. This is sort of 25, and then you're going from 50 to 1,000. So this is much higher. You can see that at the very lowest doses of mRNA, you get a drop off, and then we did another titration where we actually went down even lower. And so you can see that that the VHH3 here in teal was actually required relatively more antigen expression in order to trigger the interferon gamma production compared to the VHH1 in magenta, which required less. Conversely, this VHH2 actually kind of had a flattening off. So there was some RAPA-dependent but antigen-independent signaling of this particular clone, which we wouldn't have detected if we hadn't done this experiment. So we thought that could contribute to a tonic signaling of the CAR in the presence of RAPA, and so we excluded that clone from sort of candidacy for clinical translation. There's some ideas that maybe having a little bit of tonic signaling is good, so I'm not sure that was the right or the wrong answer. We just don't have assays to read that out, but them's the data, as they say. Okay, so this is a little bit different experiment. So that was the amount of antigen, how much CD33 you needed. So what about how much rapamycin did you need? So again, 3:1 effector to target ratios, the CARs were put in a dish, and then we added various amounts of rapamycin. And even though the FKBP, FRB are the same, we were wondering whether or not the effective concentration required to half maximally activate the T cells would be different based on the different receptor architectures. And what we found was actually remarkable consistency. So the amount of rapamycin that you need to fully activate these CARs is about 0.25 nanomolar, very low. Just for reference, in patients, when you give them rapamycin, say in the post-transplant setting, the amount of rapamycin in a whole blood assay usually is targeted somewhere around 5 to 10 nanomolar, and certainly up to 20 would be reasonable. So we're well below that here. Okay, so is there a way that we can verify that the model that we are envisioning here is in fact what's happening in this system? So this was a reviewer experiment, but I think it's kind of interesting. So we designed a FRET pair. This is a phycoerythrin Luxifer 647 pair. These have donor-acceptor overlap on the flow cytometry curves. So the idea is that in the paused state or the resting state, the PE will accept blue light, it will emit green light. The FRB 647 can, when the two arms are brought in proximity and complexed by rapamycin, will accept a non-radiative energy transfer and emit red-shifted orange light. And that's what we saw. So this is looking at the 647 channel, and you can see the This is just this is a control experiment looking at just actually anti-IgG after staining the anti-VHH, and so you can see that you can get efficient FRET between these two donor and acceptor pairs in the 647 channel, which is over here on the right side, and actually in the PE channel, which is over here. Sorry, this isn't labeled. You get decreased green emission, which is expected because this green photon, instead of radiating out, the energy gets transferred over to the 647 fluorophore. So this is kind of confirmation. When we titrated in additional rapamycin, we got a nice dose-dependent increase in the FRET signal, and this was really restricted to the dual positive cell population here. So this is the FRET signal within the PE-Cy5 channel. And we could use this FRET signal to actually measure the EC50 of heterodimerization. Interestingly, before I showed you that the maximal amount of heterodimerization happened around 250 picomolar, the EC50 of heterodimerization by interferon gamma is probably around 20 picomolar. The EC50 of heterodimerization when measured not by interferon gamma production but by FRET is slightly higher, 135 picomolar. So you probably don't have to complex every single derrick on the surface of the cell in order to maximally activate the CAR. And I guess we could back calculate what proportion of the derricks we thought we were complexing, but not all of them. Rapamycin is, yes?
Sorry, can I just clarify, is this ex vivo assays of the product?
These,
This is not a blood draw from a patient after they were.
correct.
Okay.
Correct. We are working on developing that. When I say we, I mean Stephanie Rea and CSL and Camilla and all the great people upstairs in SETX and CSL. But there we have validated. Oh, so the thing on the right is mock cell products, like clinical products that we spiked into blood from healthy donors. So that is sort of what we can get to in terms of the preclinical scenario. Can we, from blood matrix, can we evaluate heterodimerization? And the answer is yes, we can. And that was actually very useful because you can see that there's like 100-fold decrease in the EC50. So now you're up to 11 nanomolar as opposed to 0.1. And that actually makes a lot of sense because sirolimus is sopped up in red cells and protein bound and all these things. So the amount of free circulating rapamycin in the patient is not comparable to the amount of free rapamycin that you put into serum-free T-cell media.
This is a realistic
Facsimile.
concentration that you would have in a patient if they were on rapamycin.
Absolutely. Yeah. Okay, so now what we did is we said, okay, there's differential binding of rapamycin within media, humans, and actually mice. And so we wanted to say, okay, if we're going to try and have an animal model of rapamycin activity, can we do the PK? Can we compare the PK in mice to the PK in patients? And so we took out we sacrificed a whole bunch of mice and collected like 50 mils of mouse blood one day, and then Michael Baldwin did. Thank you, Michael. And then we got some human blood, and then we got some T-cell media, and you can see the variance in the EC50. So 139 picomolar, 2.6 nanomolar for both human and mouse blood. And this, again, is by activity, not by FRET. So this is the interferon gamma production. So what we did was we spiked the CARs in with tumor cells into the samples. So they would all have human CD33 to react against. And so now you have an amount of rapamycin that you need in blood matrix to half maximally activate the cells, which again is at the lower limit of what is actually detectable by a clinical assay, which is about 1 nanomolar. So we're at the very lower limit of what's detectable by the clinical assay. We looked at a little bit at the kinetics. So what's the concentration of RAPA? So low nanomolar concentrations in order to activate. What about the duration? So how long does the RAPA need to be around before the CARs are on? So this is the activation, the sort of K on. And what we did was we did an IncuCyte experiment where we took the tumor cells, took the Derek cells, spiked them in, added RAPA at time 0, and then measured the tumor signal. And so you can see that you get a nice time-dependent decrease in tumor signal here. And then if you measure the difference between subsequent time points in the IncuCyte, you get effectively a killing velocity. So this is how fast are the T cells eliminating the tumor? What's the drop-off in tumor signal? And so, and of course, similarly, if you took the derivative of this, you're going from position to velocity to acceleration. And so this is how fast the killing velocity is increasing, and it's negative because the tumor signal is decreasing. And so what you can see is actually the tumor killing velocity increases right until before you eliminate all the tumor cells. So the T cells are ramping up and killing faster and faster and faster until you eliminate the targets, and it becomes hard to find somebody to kill. And so what this suggests is that at the moment of activation, there's a little bit of a delay, and at this point, the cells become sort of kindled, and this might be at least some. I don't know that I thought about this for a while, but in terms of the clinical experience, it may be the case that we don't want to have rapamycin around for all that long because, again, the T cells become more and more active. And of course, we don't know if this is true in other CARs, but this is a feature that might be useful in terms of mapping CAR reactivity. We can prolong the survival of mice in xenograft models. So this is an aggressive AML tumor xenograft model. Mice who are untreated or don't receive rapamycin are dead within 20 days. If you receive rapamycin in this model, the survival is prolonged out to 50 days. This is an aggressive model, so we weren't expecting the mice to be cured. But it does show activity. What? These are all delivered intraperitoneal, so IP injection. And we add it every Monday, Wednesday, Friday. We meaning Kaori Oda in this case, right? Just being clear. Okay, so we wanted to design an experiment that would model a pause in rapamycin. And so so can we turn it off and turn it back on? And so what we did was we adoptively transferred an AML tumor cell line, and then we injected T cells 7 days later. And what you can see in this column is that if you don't do anything, the mice just receive untransduced T cells, the tumor progresses, and the mice have to be sacked around day 40. So this model is not quite as aggressive as the mole model that I showed earlier. Okay, so then, here's untransduced cells plus RAPA. So the RAPA alone doesn't do anything. Here's Derek cells, but they didn't get RAPA. So again, the tumor progresses with roughly the same speed. If you give continuous rapamycin and Derek cells, the mice do very well. This one mouse here progressed, and so at the end you had 4 mice that had very little tumor, but the rest did very well. Okay, so what about if you stop the rapamycin on day 14 as shown here? So that's this line here, so rapamycin up until day 14. If you do that, tumor burden goes down, you stop the rapamycin, and now tumor burden starts increasing, and all of the mice relapse except for this one lucky mouse that is right here that happened to be cured by these 2 weeks of treatment. So there's some amount of residual tumor in these mice at day 14 that if you don't administer further rapamycin is capable of inducing relapse. So what happens if you then restart rapamycin? So if you restart rapamycin 2 weeks later on day 28, now instead of 4 mice relapsing, only 1 mouse relapses. So that suggests that restarting rapamycin on day 28 had additional anti-tumor activity. Okay? And in fact, the survival of these mice was 4 out of 5, which is actually remarkably similar to the survival of the mice that got rapamycin continuously. What about dosing? So this was a big experiment. We took mice, we gave them MV411 tumor cells, we divided them into a bunch of different groups, and we added different schedules of rapamycin after the same number of either untransduced or Derek33 T cells. And on the left, you can see the mice that either got no rapamycin or got and untransduced T cells, or no rapamycin and Derek33s. And then you can see going to the right, they got progressively higher doses of rapamycin, and they got progressively longer survival benefit. Normally in our lab, this experiment would have been a failure because we look at mouse experiments as kind of binary, and if you don't cure the mice, then you didn't do a good enough job, and if you cured the mice, great, and so it's sort of an all or nothing thing. And after expending so much effort on this particular mouse model, we wanted to derive some information from it, and so what we did was we came up with a multivariate model that's a generalized linear estimating equation that essentially maps the tumor growth rate over the first 50 days to each individual mouse, and then comes up with a exponential increase, so a sort of an exponential growth rate of the tumor flux within each mouse. And then what you can do is each mouse, of course, is a biological replicate. You can map the slope of that mouse's tumor growth to the treatment. And so you can see here that the mouse that got no RAPA had an increase in flux that was about between 0.3 and 0.25 and 0.37. So this is the spread of all the mice that got no rapamycin. It was about 10 mice. And in fact, the other inactive controls are shown over here. So this is mice that got no treatment at all. This is mice that got untransduced T cells. These are mice that got Derek33 but no RAPA. These are mice that got untransduced T cells but RAPA, but did get RAPA. So you can see that these are all kind of the same. Okay, so now in the going this way, the teal was the lowest dose of rapamycin. You can see that the modeled slopes here are the lowest. So this suggested that even though we were sub-curative, the biological activity actually was more at the lower doses of RAPA, suggesting that there might be some T-cell inhibitory features of RAPA at higher doses, even though we were sort of subtherapeutic. That might be the idea. At the same time that we were doing that experiment, we actually drew blood from those mice, sent it to the clinical lab, measured the rapamycin levels in the mice to try and map a dose-activity relationship. And so these are the PK curves of these of the doses. So each of the peaks is the rapamycin administration. That's the black lines here. And so in this case, the lower limit of quantitation is about 1 nanogram per mil. There were some that had very high doses of RAPA. There were some that got up to around 10. This is kind of the lowest dose of RAPA. This is be very reasonable for a post-transplant patient to get this amount of RAPA. So we thought we were in the right milieu, I guess, the right zone of concentration. We actually modeled this as a two-compartment model. We measured the apparent clearance rates in the T1/2, which is about 3 hours, so fairly rapid off in the mice, which is more rapid than what we would expect in patients. Having these values then allowed us to translate the amount of rapamycin that we would need to the human scenario, because human rapamycin PK is well described. So now what we did is we took a population model, so among pediatric patients, what is their estimated size, what is their estimated renal function, what is their estimated total body water and volume of distribution, and if we give them rapamycin, what are the levels that we're going to get? And then we map that population sample onto like a mean and 90% confidence interval, and so the mean is the dark blue and the 90% confidence interval is the light blue. So if you take 500 simulations of 0.5 milligram daily, day 1 is this, day 2 is this, day 3 is that, and then you sort of eke out into this kind of flat, steady state eventually in the sawtooth pattern where you're around about somewhere between, say, around about 2.5 nanograms per mil is sort of your steady state, and the peak is closer to 3 and the trough is closer to 1. So that's that. So then we map these different doses, the medians of these different doses on. And so we kind of, and then this green bar here is what we think we need to actually activate Derek. So we chose the half milligram, we chose the 0.75 milligrams daily for patients that are larger, and then half milligram per meter squared for patients that are smaller. So so we used that PK model to estimate the amount of rapamycin we would administer to patients in order to get them activated. Okay, so just last couple of slides. Did this actually work? Well, we think it did. You need to model cell dose, the rapid dose, and the on duration as features that will that are all variable in terms of how much activity the patients are going to get. And so that actually is a little bit challenging. But what we decided was that we were going to keep the on duration fixed, and we were going to try and have a dose that provided full on. And then the phase 1 dose escalation was not going to be in terms of adding extra RAPA or adding durations of RAPA. It was just going to be the amount of cells that we injected. So we kind of collapsed our 3 axes into one. This is the trial. This is Plateau 8. Adam's the PI. Dose escalation is 1, 5, and 10 million cells per kilogram. Flucide lymphodepletion is standard. RAPA days 2 through 21. And then if the patients have hematopoietic recovery, we can and a response, then we can pause the RAPA and see what happens. Okay, so first 3 patients, what happened? The rapamycin level was, I would say, not achieved in patient 1. In patient 2, we measured a little bit more frequently. The successful draws where we were in the range that we wanted to be in are in black. The ones that are subtherapeutic are in white. And you kind of see that we got most of the time in the right area, 8 out of 15 draws. And subject number 4, more frequent measures, more frequent draws, 14 of 18 time points in the target range. Did it work? Well, maybe. There was one patient that had a chloroma on their forehead that underwent hemorrhagic conversion and necrosis after Derek33 infusion. You can see the lesion on the bottom here got swollen and then fell off, essentially. And so we collected this lesion and performed flow cytometry on it. And if you look in the chloroma, what you can see is there's an enrichment in the proportion of cells that express both the VHH and the FRB. I didn't tell you this before, but the FRB is actually destabilized. So in the absence of RAPA, that signaling arm is internalized and degraded. So those cells are FRB negative. And in the blood, there are a few VHH positive cells, a couple percent, but very few FRB VHH double positive cells, whereas within the chloroma, they're enriched. So I think I think this is evidence that the cells are trafficking to the site of disease. If you look at those cells, they do express increased proportions of PD-1 and TIM-3. Again, suggestive of the fact that not only are they active by the fact that they express both arms, and not only are they enriched in the sites of disease, but they express markers of activation, PD-1, TIM-3, suggesting that they've been stimulated by antigen. Now, I don't have information about their functional aspects, like are they making interferon gamma, but that might be something that could come later. We did see in patient 4, or subject 4, that the improved rapamycin targeting was associated with improved T cell proliferation in the patient. So if you just look at the proportion of VHH positive cells within the blood, that patient had an increase in the proportion around day 8 or 10. I will say that this is maybe a peak that's a little bit later than the peak that might be expected for vanilla CD19 CARs, but I'm going to go into that a little bit more So just again, kinetic-wise, in subject 4, that peak of Derrick cell expansion around day 9 was coincident with a reduction in the side scatter low CD33 high circulating cells that we presume are AML blasts. So while our typical measure of AML blasts is now high parameter flow cytometry, in this particular case we do not have all the parameters because we use parameters for T cell phenotyping, but blasts are side scatter low CD33 high. This patient had high circulating cells at day 7, so they're the cells, I'm going to call them blasts. On day 14, that proportion went from 88% to 0.23%. They came back soon thereafter, so the elimination was not sustained, but at least some transient evidence of anti-tumor activity. Anti-tumor activity was not all we had. We had a lot of cytokine production. If you look at day 10, which was again right around the peak of cell expansion, we had high levels of IL-6, high levels of TNF-alpha, high levels of interferon gamma. Interestingly, this particular subject, subject 4, also had high levels of circulating IL-15 compared to the other 2 subjects. IL-15 being a cytokine that is a pro-survival cytokine for CD8 T cells in particular. Okay, so in the last couple of minutes, just a little bit about the next generation. So that study is ongoing. I believe it's now reopened. So if you have patients or, you know, I guess I'm not talking to the patient people, but if I have patients, I will let Adam know. So there you go. Okay, IL-15. So IL-15 is a T cell survival and proliferation cytokine. If you dose monkeys with IL-15, this is done by Stan Riddell, you can raise a white count from around 10,000, which is kind of normal for a person, up to 70,000, which is too high. But if you stop the IL-15, the T cells come back down. So IL-15 has proliferation expansion effects. It preferentially expands central memory and effector memory cells at very high levels. You can get autoimmunity. You get anemia actually due to marrow infiltration, so the hemoglobin dropped all the way down to 6 in these animals. So it can have side effects. So can we engineer a couple layers of safety for our potency enhancement? So we already talked about rapamycin regulated T cell activation, but we engineered 2 other layers. One is this synthetic transcription regulation called ISIM-PRO, which stands for inducible synthetic promoter. And can we drive IL-15 to perhaps improve the expansion of these T cells as well as their function? So just in terms of, I gotta skip this. Okay, so here's IL-15, ISIM-PRO in a nutshell. This was a lot of work, but this is the idea. So we took a bunch of transcription response elements, the binding sites of transcription factors whose activity we know is increased when T cells are activated. We did a pooled ligation with end caps to generate a synthetic promoter library that we then ligated upstream of GFP in a lentiviral backbone and used it to transduce T cells with. This produced a large number of potential promoters where we had repeated elements of, say, NF-kappa B, FOS, NFAT, which had the potential to drive GFP following stimulation. So we then took those T cells, stimulated them, and sorted out the GFP positive cells, and then purified DNA and subjected them to NGS sequencing. The NGS sequencing results were then used to create clones, to purchase clones, transduce T cells in the presence of a CAR, and then repeat the stimulation. And what we found was that a large number of these sequences that we covered by NGS actually functioned well when we retested them in the setting of CAR activation. And so you can see here in 2 different donors, if you look at repeated stimulations with Raji tumor cells, the CD19 CAR cells co-transduced with these putative inducible synthetic promoters induced GFP to a high level that was transient and then returned down to baseline following repeated stimulation events. And this worked over, again, in both donors. I'm sorry that this is, okay. So what could we learn about this? Well, we took these we took these promoters and we then did a time course where we transduced CD19 CAR T cells, we stimulated them with Raji's, and then we isolated DNA at various time points afterwards. And then we figured out which promoter sequences were present at multiple time points and how much. And so so here's the first 3 time points. So 24 is this axis, 48 is this axis, 72 is this axis, and then the color is following a restim on day 7. So if you're present in all 4, you're positive, 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 and dark. So these guys up here were all 4 time points. If you were just 1 time point, that's down here or over here, depending on which time point you were positive. So this is kind of a 4-dimensional representation of the frequency of these promoters at various time points. It's a little hard to sort of see. So what we did was we actually mapped it out. So the 4 kinks in these lines are the 4 time points, and we clustered the different relative abundances of the different sequences by how much they were present at the various time points. What we found when we did this was that the abundance of the various transcription factor binding sites actually segregated by the behavior of the sequence. So if the sequence was equally present at all 4 time points, you had an abundance of, say, June. June is a well-known T-cell responsive transcription factor binding site. Makes sense. 12 is this guy, which actually induces a little bit late and then doesn't induce very well on restim. And so 12 has EGR1. So that's a different one. So there's different patterns of sequence specificity based on the behavior of the isompro sequence. And one sequence in particular, this JOU, happened to be well enriched in this cluster 2, which had early induction and then turned off and then was present on re-stimulation. And so we actually investigated this particular isompro further, and we did this in this flank model. And what you can see is that when these isompro GFP cells were injected into a mouse that had either a CD19 positive tumor versus a CD19 negative tumor on the left versus right flank, respectively, all the tumor signal was in the side of the flank that had the antigen positive tumor. So it's both spatially and temporally localizing T cell activity. Okay, so now we clone that upstream of IL-15 so that when the T cell gets stimulated, it secretes IL-15. And just gonna go, I'm gonna skip this, but this is okay, so this is the last experiment. So again, mouse model, untransduced T cells without RAPA, untransduced T cells with RAPA. This is the Derek 33. This is the sort of what we're using in Plato 8. This is the isompro IL-15 at 10 million. Again, these cells don't do much if you don't add RAPA. If you add RAPA, you suppress the tumor growth for the entire course of the experiment. But now we reduce the dose by a third. So now instead of 10 million, you get 3 million. And the Plato 8 Derek 33 only controlled the tumor in one mouse, but these 2 different versions of the enhanced IL-15 controlled the tumor in 4 or 5 out of 5 mice. Okay, so where do I think we can go from here? Well, so we've got potency enhancement, we've got controllability. What does the future hold? And so here's just a brief pitch. So we know a lot about CD19 CAR, and I think the success of CD19 CAR is potentially importantly driven by the fact that it engages its target in the context of the surface of B cells. But CD33 CAR is a much different beast. The myeloid cells are suppressive. What happens if you make a CD19, CD33 dual CAR? Does that overcome the suppressive activity? And so if you make a mixed population, you actually sort out those 3 groups, the single, and the double, to figure out is one dominant over the other, and what are the differences in that signaling pathway. So with that, I'll just say thank you. This is a lot of work by a lot of people. Appreciated the opportunity to talk to you today. I'm happy to answer any questions.
Jake, that was a beautiful talk and an enormous amount of work. I have a quick question for you. The ice and pro stuff that you showed is actually phenomenal, and then the PK analysis that you had done with the rapamycin. Have you looked at utilizing the differential promoters at these different time points and activating them with rapamycin to induce biological activity? Like, is this something that you guys are doing?
Yeah, so I think that this is an incredible tool that could be used in a lot of different ways to try and understand the kinetic relationships between functional outputs and kinetically defined genetic perturbation. And so, now you have a way of inducing, say, like what happens to a T cell if it's bathed in IL-6 from the moment it gets activated. And we haven't done. I mean, I don't think we've done that, but it would be interesting to think about that kind of model. I mean, not so much. I mean, the IL-6 is a little trivial, right? Because you could just add IL-6 to a dish, but you could do other things like induce dominant negative blocking certain signals that might be other ways of manipulating cells in interesting, yeah.
So if we were to apply like the Derek to other conditions, say beyond oncology where you want an on-off condition, what do you know about the longevity of the T cells in the absence of RAPA and antigen?
So not a lot. As I mentioned yesterday in Katie's talk, I think the mouse is a pretty broken human immune system, so it's unclear. Now that said, we did do some experiments early on with our CD33 CAR and our CD19 CAR, where we loaded the mice up with Raji cells, we treated them with CARs, and then we would retreat the mice with a second dose of tumor cells like 100 days later. And even in the NSGS or the NSG, mouse that doesn't have a lot of T cell cytokines, those cells stuck around and rejected the tumor, 3 months later. We did not do that with the Derek cells, and we didn't do it with the Derek IL-15 cells. So it would be interesting to try. That would be an interesting experiment, and that's one of those things where it's like sort of self-sorting also. So if you had a whole bunch of sequences, it's, for example, like activated different cytokine targets for transcription. You said, okay, I'm going to transcriptionally activate, 10 different cytokines, or make a viral library with 10 different cytokines. It'd be easy to figure out which one is enriched, day 7 post-tumor rechallenge to show, oh, this one not only is present but also remains functional. I think that'd be a great experiment to do.
My assumption, just from catching the 3 patients that have been treated, each of those were only treated once, right?
Yep.
And how long post? How long have we waited? Because the one patient seemed like they responded really well, but presumably, based on nobody saying anything, that patient has not, the disease has not gone to zero, right? This is not a cure in that 2-week period. Is the trial designed such that they'll go on to transplant, or is that?
Yeah, yes. So you can see here that the gray bars are the total CD33 antigen. So this is sort of the sum of all the CD33 MFIs. And you can see it kind of goes up a little bit and then goes down, goes down, goes down. And then even by 3-4 weeks, it's back up. So this patient had a very transient, like days long response. This is not the home run that CD19 CAR was.
Yeah.
And it is clear to me if that is, I mean, I want to be optimistic, but I also think that that's a signal. So, you know, again, is there something that is defective about the way that these cells are signaling? Is it the environment that they're going into? Nobody has cracked that particular nut yet, so I don't know what the answer is. But yeah,
And the pink bar for rapamycin, is that when the drug is dosed or when the drug is present in the patient?
that's when the drug was up. That is the days during which the rapamycin was above the threshold for activation. So
So it's longer, yeah.
yeah. So this patient relapsed when rapamycin was continued. Now, if you know, so by day 21. Now, if Sarkar were here, he would tell you that exhaustion is probably epigenetically programmed very rapidly. And so if you look for transcriptional changes, those transcriptional changes, at least at the single cell level, begin to be evident like at day 3. And Sarkar thinks that it's actually within the first cell division. Like if you have a T cell and it's attaching to a dendritic cell and then it buds off a daughter cell, the cell that happens to be next to the dendritic cell is the cell that becomes the memory cell, but the cell that's on the other side ends up becoming the effector cell. And that cell goes on to proliferate and make a lot of effector cells, but then that cell that is stuck to the dendritic cell that's having more like stimulation for longer becomes, you know, is kind of quiescent, is dampened. I don't know that everybody believes that, but at least it suggests that a very different clinical approach could be to give like 1 or 2 doses of rapamycin and then, you know, let them be on for like a day and then pause and let the cells recover. And there is some epigenetic data, if you look at like ATAC-seq, that first stimulation event is maybe qualitatively different than other stimulation events. So that first stimulation event, if you then re-stimulate those cells are kind of protected from further exhaustion. Whereas if you have a longer stimulation event up front, they're exhausted and they stay that way. So again, I think in order to do that, you're gonna have to be in a clinical setting where you have a little bit more room to maneuver. I mean, this patient relapsed in 3 weeks, so you can't wait for the cell to proliferate and then redose rapamycin. I mean, maybe you could, but you know, the window is short for this patient. If you had a patient that was a little bit of a deeper remission, had kinetically less active disease, so that you had a little bit longer, maybe you could, you know, dose the rapamycin, wait 2 weeks, then redose the rapamycin. That might be a possibility.
Make me run. Yes.
Hey, great talk, Jake. I have a question regarding the last pitch you have for CD19 CAR and the CD33 CAR dual. Can you talk more about that? And are we also assuming a B cell aplasia and how that would affect the CD33 CAR targeting?
Yeah, so I will say that there's a pretty robust literature of these myeloid-derived suppressor cells, where if you take MDSCs and you co-incubate them with T cells, that those T cells have blunted effector responses. And if you look at the transcriptional signatures of AML cells, they look an awful lot like myeloid-derived suppressor cells, and they look an awful lot like immature melanocytes. Those things all are kind of suppressive. It's unclear whether the CD19 CARs can provide help in trans to CD33 CARs that are suppressed by MDSCs, and it's unclear if it can provide help in cis if you have in the same cell versus in an adjacent cell. And so this would be a way to get at that. Could you have a population of CARs that just don't care about what the CD33 cells are, but they kind of ignore them? And if you start responding to the CD33 CARs, does that make your CAR effect worse or better? It's really unknown. But there's a population of patients out here who have both CD19 and CD33 on their tumors. And if you only target one or the other, there's very likely to be antigen escape. So maybe we can learn about how that reactivity works while helping some kids and adults at the same time. What do you think? You buying?
Thanks a lot. No, it's fantastic. Thanks.